All right, let's do this. Good morning, everyone. Welcome to Friday's class. Glad to see everyone here. We are going to pick up where we left off today, talking about histology. We'll wrap up histology today and move in, if we have time, to the integumentary system. But this is a perfect kind of background information for your first couple labs coming up. So let's take a look at what we're going to do today. As I said, we're going to start off with a quick bit of review on embryology that we covered the last couple lectures. We're going to finish our section on histology today, including a short drawing activity. And then we'll start into the integumentary system. We'll see how far we get. Wherever we leave off, we'll just pick up on Monday. Then I'll wrap things up by providing some details for quiz number 1 and tick it out for you today. And of course, it is Friday, so you get your Friday Venn diagram. Before we launch into it, questions? All right. Oh, one kind of logistical note. In Blackboard, for assignments like the tickets out or other things like that, I set the due date when it's like the end of class period. So for example, if we have a ticket out today, it's going to be due at 8:50 a.m. on Friday, the 2nd of September. It takes me a few days sometimes to grade those and get them in. So if you get a notification you have a late assignment and it's a ticket out, it's fine. Give it a couple days for me to do that, because I have to manually then look at all those assignments, read them in Google, like in the Google Forms, and then put that into Blackboard. But it's going to tell you you're late, but you're not late. You're cool. So just be aware of that. You can kind of ignore those auto messages from Blackboard. I don't know if there's a way to turn them off, but I find them kind of annoying. So you can just ignore them. Review. Let's talk about embryology. So in the middle of last class, I asked you to work with a partner and fill in the blanks from these sentences. In this sentence, singular. In human development, the embryonic stage is characterized by blank, whereas during the fetal stage, blank. I looked through your answers. Seems like the class has a good understanding of this. Here's one answer that kind of nailed it. So remember that the embryonic stage is characterized by the forming of the germ layers and the start of the basic organ systems. And during the fetal stage is a time of growth and further development. So all of the basic structures are formed early in the first 8 weeks, remember, during the embryonic stage, and then they continue to get larger and more refined in the fetal stage generally. So good job with that. Any questions? Kind of hammered this point home a little bit so far. You remember nothing else from the first 2 weeks of class, it's going to be this. All right, so last class we talked about tissue types, and I talked a little bit about epithelial cells. And I just want to back up and offer a little more information on epithelial cells. Actually, not more information, but the same information in a little bit of a different way with some images on these slides. And then we'll launch into the connective, nervous, and muscle tissue we didn't talk about. So just to recap, remember that epithelial tissues are defined by how the cells are distributed. So whether they're simple or stratified, so that whether there's one layer or multiple layers. And you can see some examples of this here. And I wanted to show you this. This is an image from your book, and there's more details in the book, but I wanted to just illustrate this. I talked about this in class, but I didn't really give you some good examples. So these are actual images of different human tissue types. We'll be looking at these slides in lab next week, or no, the week after next. We're a week behind now. But you can see here that the simple epithelium is just this single layer of cells you see right here, right? Now, if you had a guess looking at these cells based on their shape, would you guess that they're squamous, cuboidal, or columnar? Cuboidal, yeah, they're more or less square shaped. So these are, my guess is this is probably from a duct in the kidney, or one of the tubules in the kidney, where you have this single layer of cuboidal epithelial cells. We also have stratified epithelium. So this is when they're stacked, and you can see here all of these dark spots are the nuclei of cells. You can see a whole bunch of them stacked on each other. And we also have another type of epithelial cell that I didn't mention on Wednesday. It's called pseudostratified. Pseudo, of course, meaning fake or false. So these are cells that look like they're stratified, but they're not. And the reason they look like they're stratified is because the nuclei are kind of in different positions. Some are closer to the basal side, some are closer to the apical side. And so you kind of have this look like they're stratified, but in fact, there's just one layer of cells along here. So these are cells, for example, that are found lining the your trachea. And you'll see examples of this in lab as well. So just a review of simple versus stratified and introducing this new term pseudostratified. We'll look at that in more detail, but I just wanted to kind of set you up for with some visual images of that. Oops, wrong way. Then remember, in addition to being simple or stratified, we can also describe these cells as being squamous or flat, cuboidal, or columnar. And you can see some examples here. I'm not going to walk through all the text here in detail. This is in your textbook. But the key thing I want you to take away here is the images. You can see these squamous cells when they're layered. You can see all of the nuclei kind of on top of each other. And cuboidal cells, you don't actually see cuboidal stratified very often in the human body. There's only a couple places where you'll find that. And then for columnar, it's pretty rare, even more rare to find stratified columnar cells. You can see how these are kind of tall cells that are taller than they are wide. Yeah, welcome to class. Hey, wow, class is growing. Here, let me grab, oh, I only have one piece of paper left. Okay, yeah. Good to see you both here. I saw both of your names on the list, but I didn't actually see you. So yeah, cool. Oh, really? Yeah. Okay. Yeah, the Science Center is notoriously confusing because yeah. I like to mix things up a bit. So yeah, takeaway messages here. Again, reminder, you can classify these by their shape and then by if they're single layer or multiple layers. See some examples there. Any questions on this? Kind of hammering that point home a little bit, but it's important. Also, this is a review. This slide you saw on Wednesday that epithelial cells also produce glands, and so these are derived from the ectoderm, just like your epithelial cells are. And these are types of epithelial cells. So I shouldn't say as epithelial cells are, but some epithelial cells develop into glands. We have endocrine and exocrine glands. I talked about this a little bit on Wednesday, but I just wanted to add this diagram so you can see what this looks like. So this is an example of a unicellular exocrine gland. So this is a type of gland that excretes something, I'll just leave it at that, through an opening. And you can see basically in this case, this is a cell that produces mucin. So this is what is responsible for producing mucus, for example, in your upper digestive system. And you can see here how it has this big vesicle, this big kind of lumen, this big open area with all of these vesicles that contain mucin, and then they can kind of pop out of the top of the cell. So I just wanted to show you what that looks like, because I didn't include this diagram in Wednesday's lecture. Again, that one is from the book, so you've got that one as well. In fact, it's figure 4.4 from the book. Cool, we good? All right, let's talk about connective tissue then. I touched on this a little bit on Wednesday, so I'm gonna put this slide up. I covered it already, but just as a reminder that all of these types of connective tissues are derived from this embryonic tissue called mesenchyme. This is kind of the precursor to all the connective tissues that appear in the adult body, or well, not necessarily adult, but the body beyond the embryonic stage. So you can see what mesenchyme looks like here. It appears very early in development. And then this mesenchyme differentiates into a number of different types of connective tissue. And remember, this can include what we call connective tissue proper. I'll describe that. You'll see that in more detail in a minute. But also cartilage, blood, and bone are types of connective tissue. Remember that connective tissue generally serves the function of providing structure to your body. And this can be to provide structure to internal organs, things like blood vessels or your respiratory system, but also structure to features on the outside of their body. So for example, bone is what provides structure to your limbs or something like that. I want to talk a little bit about this one type of connective tissue proper called areolar connective tissue. This is kind of the model connective tissue. We can describe other types of connective tissue proper in relation to what we see in areolar connective tissue. This type of connective tissue underlies your epithelial tissue. So it comes into contact and is often adhered to epithelial tissue at which side of the epithelial layer? The basal or the apical side? Basal, good. Remember that basal side of the epithelial layer is the side that's facing the rest of the body, right? And often the tissue that is underlying that is this areolar connective tissue. And this areolar or connective tissue, it surrounds blood vessels and nerves, kind of travel through it. And it functions to kind of wrap around your organs and protect them. It holds these other vessels in place, the nerves and blood vessels, so they have like a steady place to go, so they stay in place where they're supposed to. Also plays an important role in some types of immune function and inflammation. I'm not going to talk about that in too much detail, but they can get swollen under infection or irritation. And so often it's these tissues that, say, for example, can get swollen to form a rash or something like that. Now, if we look at this areolar connective tissue in a diagram, it looks like this. What I want you to take away from here is that it's kind of complicated and there's lots of fibers going through. So I mentioned that there's blood vessels traveling through this areolar connective tissue. There's also nerves, which I believe, where are we at there? Are there nerves in this one? No, I'm not seeing nerves, but there are nerves traveling through here as well. We've got the ground substance is essentially like the matrix of material that surrounds the cells themselves. And so in this case, we have lots of fibers. We'll talk about these different fiber types. If you see these different fibers, so that's what's going to allow this tissue to maintain its structure, but also be kind of flexible. You don't want like a super rigid tissue, for example, surrounding your stomach, because your stomach has to distend and contract and move around. And so you want those structures to be tough, but also pliable, right? We also see types of immune cells integrated into the areolar connective tissue as well. And these serve, of course, to fight infection and remove potentially harmful molecules from the body. We'll see some slides of this. Again, we'll talk about this in more detail, but I just wanted to give you an example of one type of this connective tissue proper. We'll also see examples of bone, cartilage, and blood in the lab. That's all I have to say about connective tissue. Questions? Yep, go ahead. So, that's correct. Yep. So you'll actually, well, I'll show you this diagram. I'll do it over here so it shows up in the video. I'll show you this diagram probably later today or next week in class. But you have your epidermis, then you have your dermis, and then you'll have the connective tissue underneath it. And I can draw it, but basically you have a layer of this, a layer of this. This is the world's worst diagram, and then a thicker layer of this. I'll show you that in a slide in a second. But yeah, you've got the epidermis. Remember, this is derived from the ectoderm. The dermis and the connective tissue are then derived from the mesoderm.
So then your follicles, they go down because they have a blood supply.
Mm-hmm.
Do they go all the way down to the connective tissue?
Yeah, they do. And that is a fantastic question. We ask you to hang on to that because I'm going to talk about that in a second. I have a slide to show you and everything. That's exactly right, though. And the hair follicles derive from this epidermis layer, but they kind of invaginate down here, and you'll see they are supplied with blood vessels via the connective tissue. Mm-hmm. Great questions. Good. Other questions? Sweet. Let's talk about muscles. Very briefly, I want to introduce you to muscles. What I want to remind you is that muscle tissue is a composite. So it's composed of muscle cells, but also areolar connective tissue, for example, that forms the sheaths around your muscle bundles, as well as attaches your muscles to each other and to bones and things like that. And you'll see we'll talk about what kind of we'll talk about that in more detail in lab, but I just want to introduce that idea. Most of those muscle cells are called muscle fibers. So if you see the term, I'm going to erase this. Have an eraser. The terms muscle cell and muscle fiber are the same thing. I don't know why they call it that. Sometimes I think because they're kind of long and skinny and fibrous. So you hear that. So I'm going to make sure There we have that terminology. Muscle cell is the same as a muscle fiber. And they contain what are called myofilaments. Primarily, the two most important ones that interact to allow your muscles to contract are called actin and myosin. I am not going to get into that level of detail in this anatomy class. In your physiology class, or if you take an A&P, you know that we get into great detail about how muscles contract and the forces or the structures that allow them to do that. I just want you to know that there's these important fibers or filaments rather, actin and myosin that allow that to happen. There are three types of muscle tissue. We have our skeletal muscle tissue, our cardiac muscle tissue, and smooth muscle tissue. Skeletal muscle tissue is generally, if someone says the word muscle, this is what you think of. This is your biceps and your quadriceps and the muscles that you can control voluntarily to produce movement. Smooth muscle, on the other hand, is muscle that's found generally regulating internal organs that's involuntary. So, for example, the muscles that move food through your digestive system, those are smooth muscles. You can't voluntarily control what they do. And then finally, we have cardiac muscle that is found only in the heart. You can see some descriptions of these muscles here. They is based on their appearance in the microscope slide. I'm not going to get into that right now, again, because it makes more sense to do that when we actually look at them through a microscope slide. But you'll be able to tell those three muscle types apart. That's what I got for muscles. Yes, Zoe.
So when you scar, do you scar like on just the dermis, the epidermis part of your skin, or can you scar deeper underneath, like connective tissue? Yeah,
absolutely. You can form. So the scar, it can be, it's different in different parts of the body, but basically it's when those tissues try to regenerate, but do so imperfectly. And then you can get like other types of tissue replace the previous type. So for example, connective tissue might replace muscle tissue. And that's why if you injure a muscle and it scars, it can become stiff because it's no longer muscle fibers, it's this connective tissue. And basically it most often doesn't form back perfectly, although some tissues are better at this than other. And actually, we're going to talk about that in just a second. So for example, kind of surprisingly, your bone, awesome at regenerating, really good at healing. In other tissue types, for example, cartilage or cardiac muscle are very poor. Great question. And you'll see when we talk about the integumentary system, you'll see how scarring affects the different layers of the skin as well. That's where you most often think of scarring, but scarring can be in deeper tissues as well. Good question. Others? Oops. All right, nervous system then. Let's talk about that. So nervous tissue is, of course, part of the nervous system. And there are, well, let's just go to this. This includes the brain, of course, your spinal cord, and all of these peripheral nerves that extend to virtually all parts of your body. The main job of the nervous system is to transmit signals either from the central nervous system out to your body to tell your body what to do. So for example, if I want to move this finger, that signal is getting sent from my brain to my spinal cord, down these peripheral nerves to my finger, telling it to move. Or to send signals back to the brain. So if my finger touches this table, it's sending a signal from that nerve up through my arm into the spinal cord, into the brain. So either signals to or from the central nervous system. And it's important to note that those are electrical signals. And we'll talk about that a little bit later on. But again, you probably cover that in more depth in a physiology class. Now, what's important to note is that your nervous tissue contains two different types of cells. It contains neurons. These are the cells that actually conduct these electrical signals. These are these are the cells that get all the credit for transmitting that information. But the vast majority of your nervous tissue is actually composed of another type of cell called neuroglia. These are all of the other cells that basically let the neurons transmit their information efficiently. So they nourish those cells, they provide nutrients, they clean out waste products, they insulate those neurons because they're electrical signals. So if they're insulated, that signal can actually travel faster, just like a wire is insulated to prevent the electrical signal from dispersing, and they serve to protect those neurons. So we have two types of cells in the nervous tissue, neurons and neuroglia. We'll talk about the different types of neuroglia and the different types of neurons upcoming. So I think, Abby, do you have a question? Not yet. Yet you will. When is the nervous system? About midway through the semester, we get to the nervous system. Go ahead, Sophie. It can be clearing out neurotransmitters. It can be other stuff that builds up, like waste products. Well, look at this guy. Look at this house centipede. Oh, I love these guys. They're so cool. All right, let's go. Oh, here. Whoops. They're we find them. I love those guys. Okay. Sorry. Distraction. Anyone got a spare HDMI cable on them? Oh, yeah, I've got one back up in I know. I think it's like got it just all right, let me do one thing. I'm gonna just pause recording and I'm gonna share my screen. Get this back up and running. I'm going to come back to nervous tissue in a second, but I am going to zip ahead. This is going to be a little weird, but I'm improvising here. Activity that I want you to do. I'll take you a couple minutes, and while you're doing that, I'm going to go grab a new HDMI cable. So bear with me on the little screen. But what I'd like you to do is to recreate this table in your notes and draw a picture of each of these cell types, and we'll upload this as part of your ticket out today. So just create this simple 3-column table and draw a diagram of simple cuboidal, stratified squamous, and simple columnar cells. Remember to label, I'm going to actually add this, label the apical and basal sides of each of them. So one more time, just to make sure you can see this. Simple cuboidal, stratified squamous, and simple columnar. Does that sound okay? While you're working on that, I'm going to set the timer for 2 minutes and see if I can make it to my office and back in 2 minutes. Yeah, come up and see if you can't see it. I'm sorry. There you go. All right, I will be back very quickly. Way past 2 minutes, huh? All right, how did drawing go for everyone? Got it? Enough time to do it? Cool. All right, let me just pick up this one. Oh, adventure. And maybe the thing broke. Now we're really screwed. All right, we're back in action. Let's get this back up. All right, this is all recorded as well, which is really great. I think that's working. All right, we're going to go with this and hope that that's working. Okay, so apologies for that. Thanks for your patience. What I'd like you to do, let's see, we talked about neuroglia, and that's about all I want to get to with the nervous cells. I'm going to just give you some general overview of the capacity for regeneration of the different cell types. And so this, Zoe, gets back to your question. Some of these tissue types are really good at fixing themselves if they get damaged. So that includes your epithelial tissue, which you would expect. You need those cells that are in constant contact with the external environment to be able to repair and replace themselves quickly. Bone is really good at healing itself. Areolar connective tissue, some other types of connective tissue, one called dense irregular, which we'll learn about in lab, as well as the connective tissue that forms blood. Some tissues are kind of intermediate. So for example, your smooth muscle tissue, your another type of connective tissue called dense regular connective tissue. Some of them can heal, but they're not great. For example, your skeletal muscle. So any of you are athletes and had a sports injury in which you pulled or tore a muscle, you know, it takes a long time to heal. Cartilage also is generally not generally, is avascular, so it doesn't have a great blood supply. That's why injuries to the cartilage of, for example, your knees can be so detrimental and take such a long time to heal, or in many cases can't heal and requires surgery. Other tissue basically can't heal at all or very, very little. So cardiac muscle and nervous tissue. And this kind of stands out because those seem like extraordinarily important tissues, right? Your function of your heart, really important. Once that muscle gets damaged, doesn't really repair itself. And so that's going to cause long-term problems. And then also nervous tissue, your brain cells are very poor at regenerating, if they can regenerate at all. They can a little bit under certain circumstances, but generally those neurons in your brain, once they're damaged, boom, that's it. Go ahead, sir.
So for those tissues that feel as if because they're more complex than the other ones, or just they don't harm?
Great question. What is the characteristic of them that prevents them from healing? I don't know exactly what. Let me think about that for a minute. What would it be about those tissues? So it can be different things in different circumstances. So for example, cartilage is bad at healing, often because it's avascular. So it just doesn't have a blood supply to like basically provide the nutrients to help it repair itself. Something like nervous tissue is rather well vascularized. So I'm not sure exactly what the mechanism is that prevents it from regenerating. Let me get back to that one. That's a good question. Other questions? Yeah, Kylie.
Nervous tissue does not heal. What would like say a traumatic like stop?
Yeah, so the brain is we're barely going to touch on the nervous system in this class because it's its own. There's its own classes just on the brain function. But in general, even as the cells themselves don't regenerate well, the brain has a great capacity to basically reconnect different neurons to restore functionality, even as it can't replace those neurons. And so that's why, for example, a stroke injury, people can recover and heal from a stroke, but it's often very difficult and takes a long time because what's happening there is those networks, those connections of neurons in your brain are essentially being rewritten, and different pathways for information are being reconstructed. And so it can take a long time. So the brain, the brain can heal in the sense that it can restore functionality, even if those same cells aren't replaced and regenerated and repaired. Basically, the brain figures out, for a lack of a better way to say it, the brain figures out a way to do what it needs to do, even with cells that are damaged or not functioning to full capacity. Yeah, something like that. And that's why brain injuries take a long time to recover from. Right? That's it.
Do that rarely, because I've heard that they can change your neural pathway in your brain. Is it like, of course, it can be people saying
no, I've actually recently read a bunch of stuff on mushrooms and what they do, the brain and other hallucinogens. I'm not an expert in this at all. This isn't my area, but I'm just interested in it. There's a cool book called what's it called? Oh, How to Change Your Mind by Michael Pollan, who's kind of a reputable science writer. That's a whole other thing. Just touched on it briefly. What it does is it basically, mushrooms specifically, they inhibit the part of your brain that tells everything else to stay in its lane, right? Your brain receives and processes much more information than you're conscious of. And its job essentially is to filter that out and just give you what's important. What those drugs can do in many cases is disrupt those mechanisms that decide what's important and what's not, and that keep different parts of the brain functioning in their little lanes, and allows like integration across parts of the brain that aren't normally integrated. There's a lot more to it than that, but that's generally what's going on there. So basically it creates kind of a neurological free-for-all in there. There's a lot more to it than that. You should ask, so in the like Dr. Vadney or Dr. Bailey in the psych department, they can tell you a lot more stuff about the brain. I'm just giving a very anatomical version of this, but there's a lot more in terms of like neurofunction. Yeah, take those classes. Has anyone taken those classes? Like, yeah, and you get into that. Right. Dr. Vadney teaches behavioral neuroscience. Who teaches the clinical one? Oh, Vadney does. Yeah, cool. Yeah, take those courses. Yeah, who teaches that one? Okay, yeah, I'd actually like to take that class. So yeah, if I say anything that doesn't match up with what you learned in that class, go with what you learned in that class. Does anything I say not match up with what you learned in that class? Okay, cool, good. That's good to know. Cool. All right, let's all right, just one more bit about tissues aging. As you know, your body changes as you age. And so by aging here, I mean from adulthood onward. And specific things that can happen is your epithelial layers get thinner. So this is why older folks have thinner skin and can bruise or cut more easily. Your collagen decreases. This is why your skin gets wrinkly and saggy. This sounds fun, guys. Something to look forward to for everybody. Your bones, muscles, and nervous tissue begin to atrophy. So this is why maintaining physical exercise is so important kind of throughout your life, because it becomes harder and harder, for example, to gain muscle mass as you get older. And then in many cases, poor nutrition or poor circulation due to a number of different conditions can exacerbate these things that happen inevitably with aging. So basically, the takeaway message there is that in aging, all of this functionality can decreases over time. And that shouldn't be surprising, but I want to talk about some specific aspects of that as it's related to these types of connective tissue. Then we have a review activity, drawing cells. Oh, you guys did that. That's great. Awesome. Let's keep moving on then. Let me pause here and see what questions you have about this histology unit, and then we will talk for just a few minutes about your integumentary system. Questions? We good? Do I want to start this? Yeah, let's just spend a few minutes just introducing the integumentary system. So this includes your skin plus its derivatives, meaning those structures that are associated with or emerge from the skin, including your fingernails, your hair, your sweat glands, and your sebaceous glands. And we'll talk about those glands in just a minute. And so I'm going to just offer a brief introduction of the integumentary system. We're going to walk through the layers of the skin, including the epidermis and the dermis. We're going to talk about what's called the appendages of the skin, so hair and nails, and then I'll just summarize things. This unit is very short. We're going to spend maybe a full lecture class. We'll probably finish this up on Monday on the integumentary system, but it is important to cover. And the first lab on Tuesday next week, you'll be looking at the integumentary system of your cat, specifically by removing it. So let's talk about your skin first, and the layer that underlies your skin called the hypodermis. So the skin, you've probably all heard this fact, is your largest organ, accounting for about 7% of your body weight, but that of course varies from person to person, but around that much. Importantly, the skin varies in thickness. So you have some places where the skin is very thin. So let's take your eyelids, for example. Other places where the skin can get quite thick. So 4.4 millimeters. So that's basically half a centimeter. It's a pretty decent thickness. So like the bottom of your foot, for example, has much thicker skin. We can divide the skin into two layers, the epidermis, which is this stratified squamous epithelium. We talked about that. And then underlying that, and derived importantly from your mesoderm, the mesoderm layer is what's called the dermis. And this is composed of two types of connective tissue, loose areolar and dense irregular. Again, I know I'm throwing out these different types of connective tissue here. You'll see these in lab. For right now, I just want you to know that the dermis layer is composed of connective tissue. That's just fine. And then deep to the dermis, we have this other layer called the hypodermis. It's not technically part of the integumentary system, but we're going to talk about it a little bit just because it is associated so closely with the skin. Now, the hair and nails, what we call the appendages of the integumentary system, develop before birth. As you know, babies are generally born with hair and fingernails. But this happens, interestingly, during the fetal period. So we talked about how during the embryonic period, all those basic structures are laid down. The ectoderm forms the layers that form the skin. And then later in development, the hair and nails develop from that layer. And there's actually different types of hair that develop prenatally. And we'll look at those in just a minute. But it's not the same structure that you have on your heads right now. I'm going to stop here, I think. We'll pick things up with the hypodermis next Monday. I do have a few announcements and a ticket out for you. But before I do that, questions? All right, oops, too far. Okay, so quiz number 1. See your class program for Monday for details, but just quickly, it opens after class today. It covers material that in our first 3 classes, so last week and then through Wednesday this week. And it includes multiple choice and short answer questions. It's open book and open note, but you need to work on your own. Please don't share information with each other. I designed it to take about 20. If you took it in class, I'd give you about 20 minutes. In Blackboard, you have a 3-hour window to complete it. So time should not be an issue for you. But just note that you can't start and stop it. Once you open it, you have to complete it in 3 hours. You can't like pause it and come back to it later. It's worth 30 course points, and it's due by Monday, the start of class on Monday, so 8 a.m. Monday. Remember that Monday is a holiday for all of the United States except for Ohio Wesleyan University. I don't know why we don't have off Labor Day, but we don't. It's a regular day. Yeah, so I'll see you Monday morning. Just a couple of notes. As I reminded you on Wednesday, please use only class material as your references. You're welcome to use anything you want, but if you start Googling answers, especially for the long answer questions, you might not get anything really relevant or just get confusing information. Everything you need is in the class materials. And by that specifically, I mean your notes from lecture as well as your textbook. Also, please follow directions carefully. One thing that comes up a lot, if I ask you to write an answer in complete sentences, please write it in complete sentences. You'll lose points if you don't. Just want to make that really clear. I'll say in the instructions, write in complete sentences. Please don't gloss over the instructions. Just as a pro tip for doing those kinds of long answer questions in any exam, read the question, then answer it. Then after you've answered it, go back and reread the question and say, okay, did I do all of these things that the instructor asked me to do with this question? And that will help you be awesome. We'll leave it at that. Questions about the quiz? Cool, we've got 5 minutes left, which is perfect. So remember your class program for Monday. In addition to the quiz, there are some readings for class on Monday on histology. We'll be kind of revisiting these over the next week or so. But note that there is a program. I do have a ticket out for you that has a couple quick questions in uploading your image from your drawing today. And as promised, here is your Friday morning Venn diagram. Go ahead and finish the ticket out. I'll be happy to answer any questions as you work through. Otherwise, when you're done, we're good to go. Have a great weekend, and I'll see you Monday morning.